Okay, thank you very much. First of all, thank you for the organizers to inviting me to this meeting. Because one of my topics today is sirtuins, I cannot avoid to talk about caloric restriction, although I am not the subject of caloric restriction. So one of the first study which came out on sirtuins actually suggested that if yeast were subjected to caloric restriction, there is an overexpression of SIR2 gene which encodes proteins. And when they ablated the gene, they found a decreased lifespan. When they overexpressed this gene, they showed increased lifespan. Therefore, they suggested that at least in this system, SIR2 protein is a limiting component of the longevity. Although I have to mention that this study was repeated recently by a German group and published also in the Nature, and they found out that overexpression of this gene did not result in increased lifespan. And first of all, I try to talk a little bit about the structure, very conservative structure of this protein. The small loop, which you can see here, small domain with zinc. Zinc actually is not really related to the catalytic activity of this enzyme. Although in case of mutation, if zinc is not present, the whole structure is collapsed. So zinc is very important. But if you see the catalytic site here, one side, you can see acetylated lysyl peptide binding point, and you can see another side of NAD, and the catalytic process takes place in this clave. And if you see the end products, of course, you will have, after deacetylation, you have the deacetylated lysyl peptide, and then you're going to acetylate ADP ribose, and again, you are going to form nicotinamide. Nicotinamide is extremely important because it has been pointed out by Sinclair group that nicotinamide is actually an inhibitor of, very specific inhibitor of sirtuins. Therefore, actually, if you have high activity of SIRT, you are producing a lot of nicotinamide, which then can inhibit the enzyme. Therefore, it's very, very important to get rid of nicotinamide. And of course, there is a process to do that, and nicotinamide can form to nicotinic acid and then NAD, and this process seems to be extremely important in order to avoid the inhibition. So there are a lot of questions because we know a lot about caloric restriction, but it is also suggested that one of the beneficial effects of caloric restriction that it increases the mitochondrial biogenesis. Of course, if you have more mitochondria, you have lower level of oxidative stress, you have better energy utilization. So there are quite many papers on this side really suggesting during caloric restriction more mitochondria formed and so on and so on. Although this claim has been challenged recently, and the group of John Holosy and his co-workers actually suggesting that caloric restriction actually does not induce mitochondrial biogenesis. And then another paper published quite recently actually shows that caloric restriction actually preserves mitochondrial function in senescence without increasing mitochondrial biogenesis. So there is some contradiction in this process, whether caloric restriction actually induces mitochondrial biogenesis or not. And then the other problem is whether the activity of PGC1 alpha is modulated by deacetylation or acetylation. Of course, if I ask you, most of you are going to say that PGC alpha activity is increased by deacetylation. This is a paper which actually shows that if you use AMPA activator ICAR, you can increase the deacetylation level, or you can actually increase the deacetylation of PGC1 alpha. You can see here acetylated lysine and this ICAR. This is time frame. So after 12 hours, almost completely disappeared the acetylated lysine here, and this is an inhibitor, which actually really shows that if you activate AMP kinase, maybe you are activating the deacetylases which activates or interacts with PGC1 alpha. This is true for myotubes and also true for skeletal muscle. And also this group showed that if you use this AMP activator, actually you are increasing the transcriptional activity of this PGC1 alpha everywhere. So this paper actually suggests that deacetylation increases the transcriptional activity of PGC1 alpha. On the other hand, if you carefully see the paper of Nemoto and his co-workers published at GBC, I just cite the two first sentences of the discussion. You can see that our results demonstrate that sirtuin and PGC1 alpha physically interact, and that sirtuin can regulate PGC1 alpha acetylation by both in vitro and in vivo. This is fine so far. In the context of KLF4 fusion protein, sirtuin appears to be inhibit transcriptional activity of PGC1 alpha. So this is completely opposite that we have seen in the former paper. Moreover, if you see another paper published by Puig-Server et al., and actually you can see this group actually, which first identified PGC1 alpha, they showed that if they acetylate PGC1 alpha, you can see CBB P300, the transcriptional activity of this transcription factor increased by tenfold. So it means actually the acetylation is activating the PGC1 alpha, and other groups are saying the deacetylation of the activator factor. So there is some contradictory data on this part. How about exercise? Exercise is much more simple than calorie restriction because you have a very nice metabolic demand here. So we know that exercise causes increased mitochondrial biogenesis, knows from the 60s, and we try to figure out whether sirtuins are playing a role in that. Actually, if you try to check the literature, there are quite many data on sirtuins and aging, but if you try to find out on rodents, on human, there's just very few data available. So what we found here, we have young control group, young exercises, rat study, old control, and old exercise group. We have found actually exercise training induced the activity of sirtuin in young and old animals as well. And when we checked the PGC alpha, actually we found that aging decreased the PGC1 alpha level, which is maybe normal, and exercise training could attenuate this decrease, and we couldn't see a significant difference between the old exercise and young control group. So this is a very nice effect in case of PGC alpha. When we checked AMP kinase, we got very similar data in case of AMP kinase activity than PGC1 alpha. So no question, AMP kinase in this model, of course, naturally plays a very important role at the activation of PGC1 alpha. We also checked some of the proteins which are involved in mitochondrial dynamics, fusion and fission. And in case of mitochondrial fusion and fission, you got an increase when the proteins are when mitochondria is accumulating maybe oxidative damage in the mitochondria or also in proteins. What we have found that in both situations, with aging, we got increased fusion and fission, and which both was attenuated by exercise training. So we got a very nice effect of the training in this part. And we were lucky to check the protein content of lone protease. Kelvin forgot to tell this, but this enzyme also activated by exercise training, but you can see with aging we got a very nice decrease in the protein content, and exercise training prevented the marked decrease. And if you see the relationship between fusion and fission and lone protease, you can see an inverse relationship, so which maybe could suggest that of course with aging you got more oxidized protein, more damaged protein in the mitochondria, and this could be partly due to the reduced activity of long protease, and which could be partly compensated by physical exercise. And a very similar study, here we checked also the activity and content of sirtuins in the cytosol and in the nucleus, because of course we could see some significant differences here. Again, what we have found that with aging we got decreased activity, and exercise training increased the activity in the cytosol, and also increased the activity of this enzyme at the nucleus as well. So it's quite simple that exercise induces the activity of SIRT1 in this situation. And we also checked NAD and the NAD content, and also the enzyme which converts NAD back the nicotinamide back to nicotinic acid and then NAD, and we found that NAMT concentration decreased significantly with aging, and exercise training prevented this decrease. Therefore, it could very nicely eliminate the inhibitory factor of SIRT1, which is accumulating as a result of the activity of the enzyme. Sirtuins are implicated also in DNA repair, and then we checked the OJG1, which is an important repair enzyme of atoxo-G. We checked the content, checked the activity, and the relation to SIRT6, because SIRT6 is implicated at the deacetylation of Q70. And we also know that OJG1 activity is also activated by acetylation. And we couldn't see any relationship between the activity of content of SIRT6 and OJG1. We have some data from human skeletal muscle as well. In this study, we have a young sedentary subjected to single bout of exercise, young physically active subject. They were also subjected to single bout of exercise. We have get the same setup for old individuals, old sedentary subjected to single bout of exercise, old active, and so on. So what we found measuring the expression of SIRT1, SIRT3, and SIRT6, in all of the cases we have found that exercise is very meaningful to activate the expression of these sirtuins. This is actually in accordance with the paper which is published from Chris Liebenberg laboratory, in which they got young subject, old subject with physically active and old subject physically inactive, and you can see also those proteins which are involved in mitochondrial dynamics, fusion and fission. They are all show some changes, and you can see that physical activity showed quite nice relationship with the young sedentary group. So exercise prevented the age-related loss in these proteins. So, and then there is a paper just coming out at Theoretical Biology Medicine saying that SIRT3 plays quite important role at mitochondrial biogenetics and biogenesis. And this paper actually shows that if you incubate SIRT3 with hydrogen peroxide, you activate or increase the activity of the protein content of this enzyme. And then this turns out that this enzyme can deacetylate FOXO3, and this group of people actually suggested that then FOXO3 further can activate PGC1 alpha. So this is another loop. So first SIRT3 deacetylates FOXO3, and FOXO3 then will activate PGC1 alpha, and therefore this is the process how you will get increased mitochondrial content. We are using also a brain as in our studies. Here we measure the DNA damage at hippocampus. We have young control, young exercise trained induced actually got a IGF-1 treatment, old control, old exercise, all the exercise with IGF-1. Here we just showing the DNA damage data, which is increased with aging, not a surprise actually. Then we checked the content of OGG1, protein content, which increased a little bit, maybe try to compensate the oxidative damage. But when we checked the acetylation of this enzyme, very surprisingly, we got a very marked, significant decrease with aging. So it seems that as a result of aging, the acetylation level of this enzyme drops significantly. As I mentioned to you earlier, OGG1 activity can be induced by acetylation. We got similar data in human skeletal muscle, the same set of that I mentioned earlier. You can see single bout of exercise actually induces oxidative damage. Not surprisingly, also in aging, you can see increased oxidative damage in sedentary groups compared to young. So this shows very nicely that aging, of course, is increasing the oxidative damage in the DNA, and single bout of exercise also pretty dangerous because there's a very limited time to adapt. We checked OGG protein level, we checked acetylation level, and then you can see that acetylation, there's a very significant decrease. So why is that? What's happening? And then we used another model. It's a brain model. This paper hopefully will be accepted quite soon. In this model, we have a very interesting set of rats. We have a low running and high running capacity rats. They were selected depending on the running capacity. So we have a low running capacity animals control. We trained low running capacity animals. We have high running capacity control, and we also train them. Just a few sentences about low running capacity. These guys are very sensitive to metabolic syndrome. They have a shorter lifespan. They are prone to different kinds of diseases. So we checked different stuff on that. We measured brain function, which was lower in this group. But when we checked the acetylation level of OGG1, actually found out that the acetylation level is decreased. But when we checked together with SIRT1, we found a very inverse relationship. So then we used a cell culture to try to find out whether SIRT1 is a potent deacetylator of OGG1. And this is what we have found. We have PC12 cells. We also treated them with NGF1, neurogrowth factor here. So when we use resveratrol, we significantly decrease the acetylation level, really suggesting that SIRT1 could play a very important role deacetylating this enzyme. When we blocked SIRT1 with nicotinamide, you can see the acetylation didn't change. Another blocker of histone acetyltransferases, deacetylases. And then when we silenced SIRT1 or SIRT3, you can see also the level of deacetylation is changing. So our suggestion is that SIRT1 is a negative inhibitor of OGG1, negatively inhibit the activity of this repair enzyme. So why is that? Is it good or bad? This is almost on the slide. We know that OGG1 can cleave 8-oxo-G, but when it's cleaved, it can form a complex. When it forms a complex, actually, it could activate cell signaling, which actually results in increased generation of reactive oxygen species. So many times, it seems, it's better not to cleave 8-oxo-G, because if it's not cleaving, maybe you will not have a serious problem. Actually, it has been shown that OGG1 knockout mice have normal lifespan, no increase of tumorigenesis, and actually they are more resistant to inflammation than wild type of rats. So this is a concept in which we actually maybe could show that SIRT1 deacetylates OGG1. So actually, our summary, we are not sure what's happening with the caloric restriction. So there are data, contradicting data. We are not completely sure that SIRT1 story is involved at caloric restriction induced, if it's induced mitochondrial biogenesis. it is also a question whether PGC1 can be activated by acetylation or deacetylation. It's quite simple. It seems that exercise very nicely activates SIRT1 and SIRT3, and this could be very important, maybe at mitochondria and also for metabolic processes. Sirtuins could be involved in exercise-mediated quality control of mitochondria, and it seems that SIRT1 suppresses the activity of OGG1. And this is the final slide before the flood. Maybe tomorrow everything will be underwater. You know, we are suffering a very serious flood of the Danube. And these are the guys who did the work. And we have a collaboration with American friends and Japanese friends. Thank you very much for your attention.
Anybody is ready for discussion? Question?
Yes, I was interested by your proposal that the repair of heterocode could be detrimentous by producing reactive oxygen species. But basic sites usually are well repaired. No? How you can, you take in consideration that we have several types of enzymes that are taking care of a basic site because they are highly mutagenic as well. So how you can reconcile, this situation?
You are completely right. Of course, DNA repair is extremely important for a cell. But maybe the fact that guanine is the more prone to oxidation also means something. So it has actually it attracts radicals. And if you see 8-oxo-G, really not really hurts the system. So of course it must be repaired, but maybe this is the damage which is less dangerous. And maybe this is the reason why guanine is so sensitive to attract radicals.
I think 8-oxo-G has to be repaired. There are different possibilities. Replication, you have a mismatch repair system. It is highly sophisticated. So, we are, okay, it's a complicated story, but I am really surprised that a basic site can generate indirectly reactive oxygen species. So this was just my comment, okay? But
okay, thank you very much.
Nice story. I just wonder, do you know anything about the mechanism of SIRT upregulation? Because exercise training and PGC1 overexpression can also upregulate SIRT1s, and SIRT could then influence PGC1. So which one comes first? And how it's regulated?
I don't know. I think AMP kinase could play a very important role. We have a paper in which we supplemented resveratrol, and we also use this low running and high running capacity model. And pretty interestingly, in one case, resveratrol induced PGC1 alpha, AMP kinase, and in that model, SIRT1 also was induced. Not in another model. And there are some other data as well. So it seems AMP kinase could play a very important role by the activation of sirtuins. But I guess other pathways are also present.
So, really great talk. Thank you. So during exercise, how do we change the level of cellular NAD?
Of course, NAD level theoretically goes down during a single bout of exercise. On the other hand, if you do exercise training, I would say you will change the NAD and would increase.
What happens to the lactate pyruvate ratio in exercise?
Well, it similarly moves NAD and NADPH. Yeah, NADH. Yeah.
Yeah. So glycolysis.
Yes. Yes.
Thank you.
And one point is, when you define a young and old, where I am.
Okay. You are young, of course. You are young.
what is all, what is young?
Okay, okay.
What is exercise and what is not exercise?
Okay. In this case, we use 30 months old Vista rats. 30 months, I think we can regard as an old model in case of rats. And exercise training, it was 12 weeks of training with 60% of the VO2 max. And we always change the intensity of the training. Every second week, we measure the VO2 max and then increase the intensity according to, so 60% of the VO2 max, what we've used.
One comment regarding George's question. The mechanism we found by which sirtuin 1 is increased with exercise is because it affects the lactate pyruvate ratio and therefore NADH or NAD ratio. And anything that will change that ratio in our hands at least increases the sirtuin expression. So at least the expression, not the activity, in our hands with alcohol or anything that affects that ratio will increase sirtuin, always towards the more lactate or pyruvate, so like in exercise.
Yeah, you're right, but it depends on the, if you use a very serious oxidative stress, of course you decrease NAD level. At the same time, you can also decrease the SIRT1.
Exercise is just lactate.
Yeah, okay. So as I mentioned, the level is important. Moderate level of decrease, of course, is an activator, I would say.
Sirtuin 1 has different substrates, and among them is P53. So if there is an increase in the activity of SIRT1, which is the effect on acetylation of P53, and if we have a decrease in the activity of OGG1, and a decrease also in the activity of P53, perhaps we could have, let's say, non-beneficial effects of activation of SIRT1, especially by resveratrol and other antioxidants.
Yeah, I think this is true for everything. The hormesis curve is also true. So sirtuins could be dangerous. In case of tumor, for instance, they are dangerous if you activate them, depending on the phase, of course, the tumor. So this is also true for apoptosis controlling. We try to always measure the acetylation of P53 because there are very few antibodies which available, but acetylated P53 is available.
So, a big question.
Sorry, did you test different exercise types and protocols in your model, and if so, do they have different effects?
Sorry, excuse me?
Did you test different types and protocols of exercise, and do they have different effects?
Actually, what we are using is an aerobic exercise program at the moment. So we don't know what happens with very high intensity exercise. There are some data on the literature, although, that high intensity interval exercise increasing the activity of SIRT1 also very well.
So whether you require deacetylation, PGC1 alpha, that's not the actual transcription factor, it's ER alpha and R1 and R2. And you did not measure actual mitochondrial biogenesis where that actually occurred. I'm asking that because there is a discrepancy with the next slide where you showed this imbalance in mitochondrial dynamics, and that's very difficult to tie up whether there is biogenesis or not.
Okay, thank you. What we measured, maybe I haven't shown you, we measured mitochondrial DNA content, we measured the activity of citrate synthase, COX, and so on. So by this, we try to evaluate the effect of exercise on mitochondrial biogenesis. Maybe mitochondrial DNA content could be a good marker.
The ratio.
Yeah. Okay.